Okay, hello, this video is going to be on developmental networks and pathways. So, our example organism for this problem is that we are going to have the spiny beetle, and the spiny beetle has a pathway which prompts the formation of spikes on its ectoderm. So in its ectoderm cells, we have the formation of spikes that make him the spiny beetle. So, to break down this pathway, we're going to start from point we really care about with the phenotype, which is the creation of spikes. So we have E induces spikes. Then we have K inhibits E. Therefore, if K is inhibiting, there will be no E. So spikes will not form. Then we have I inhibits K. So if I is present, K will not be able to inhibit E. Therefore, spikes will be generated. We have P induces I, meaning P is going to be necessary for the inhibition of K in order for spikes to be generated. And then we have S induces P, so S is also necessary for the production of spikes. So we can boil this down to S, P, and I are required for spikes to be present, as well as E, because E is producing the spikes. E is the direct inducer of the spike formation. So we have a couple scenarios where mutations could occur. So firstly, if there's a P mutation, well, if there's a P mutation, we know that P is going to be minus. This induction is not going to happen. So therefore, we're not going to have I, we're not going to have any inhibition of K, so we're going to have K inhibiting E, and there will be no spikes formed. Then we could have an I mutation. I minus mutation means there will be no inhibition of K. Once again, K inhibits E, no spikes are formed. Then we can have an I and E double mutation. Well, we know that if we have an E mutation, there will be no induction, no spikes will be formed. I doesn't really matter, it's farther up the pathway. It's not really affecting the fact that there will be no spikes because E is mutated. So then we have finally an S and K double mutation. S minus means we're not going to be producing any of this whole part of the tree all the way down to I. So that's all gone. But that doesn't matter this time because we also won't be producing K, which means there will be no inhibition of E to start with. So all of this part of the pathway doesn't matter, and you're going to have spikes. So some questions. Why don't all ectoderm cells have spikes in them? Because we have observed in this organism that not every single ectoderm cell has a spike. Well, there could be a maternal effect that occurs when eggs are created. We could have an incomplete pathway. Perhaps there's more to this pathway that we haven't seen yet, that we don't know about. And perhaps there's environmental influences. We could have effects from nutrition that affect spike generation, hormones, temperature, fluctuations. Maybe they need a precise level of humidity. It could be a number of different things. I hope this video has helped clear up pathways for you. Have a good night.